When so many target regions are combined, of course, it is important to have some kind of balancing of those capture oligos, while the approach of whole exome sequencing is definitely to cover all known exons. The main emphasis is also to cover regions that are known to play a role in human disease. Therefore, the known databases containing such variants were all used by Roche to be sure that the content there will be part of the enrichment oligos and therefore present in the sequence data that you produce, especially HGMD, for example, RefSec, but also ClinVar, Intronic, for example, and even those contents were actually used by Roche to be added to the content. So even if a known pathogenic variant would be Intronic, you could still cover it with the exome analysis. For HyperExome, we have a product where this balancing was done with really great performance. So we are really happy with how the exonic sequences are covered in this product. The on-target rate to the exonic sequences greater 80% is very typical, which is also nicely for cost-saving sequencing because you don't get a lot of off-target sequences. Our newest addition in the lab is the Roche AVENIO Edge instrument. From the feedback I get from our technicians is that it's almost too simple to operate. It uses cartridges for almost everything. They are color-coded with the reagents and also bar-coded, so the instrument can check actually its correct loading and positioning. And once this is done, a test run with the scanner actually makes sure that everything is loaded in the way the instrument expects it to be loaded. To avoid errors later in the run, you get nice signal lights, actually like a green, meaning everything passes and can start. For the future, actually, I think we would definitely like to replace our Hamilton robots with such a more complete system that can do even the quantification on deck. Currently, with the Hamilton robots, we do that off deck. And since we do 20 plexes for hybridization, we do that also manually. That exceeds actually the recommended amount that Roche recommends by a few samples, but it's really working in our hands greatly. This is a really off-hands approach of being able to do that. We are so happy with the HyperExome that we have switched our NGS workflow completely away from panels and use the exome as a backbone for virtual panels. The even distribution of coverage allows for a nice CNV detection. In this case, this was a single exon, heterozygously deleted, only one allele left. By just looking with your eye, you hardly see a difference in coverage in the upper individual compared to two randomly selected normal individuals below. However, we were able to confirm this deletion that our caller actually clearly called by MLPA. So even a single exon can be detected in a CMV analysis. However, we would always do MLPA confirmation if available for single exons. For 3 consecutive exons, we feel confident enough to be able to call them without further confirmation. Exome analysis replaced previous panel or single gene analysis. We do one streamed workflow in the lab for all these kind of analysis. Our tree analysis really has a high yield